Hello and welcome to Nikolai's Genetics Lessons, and today's problem is a tRNA with anticodon 3' end AUG, 5' end would carry, and you have to choose your answer out of these 5 answers. As usual, I recommend you to pause video here, try to solve this problem on your own first, and when you would be ready, you can run video again and you can compare your answer with my answer and explanation. So, in order to solve this problem, let me first explain transcription process. Imagine that this is double-stranded DNA, and here is another strand of the DNA, and this is transcription bubble, and here we have messenger RNA transcribed. Why I'm using different colors for this strand of the DNA and for this strand of the DNA, and why messenger RNA color is the same as this strand of the DNA. So according to this picture, as you see, this have to be template strand of the DNA. So messenger RNA is built using template strand of the DNA, and this strand of the DNA we call coding strand of the DNA. So where we can put prime ends for these strands of the DNA, it is very easy to find if you know that messenger RNA always grows continuously, and RNA and DNA grows continuously if it is grown from 5' end to 3' end. So if we know that this strand of the messenger RNA have a 3' end here, that means that here this strand of the DNA would have 5' end and 3' end here, and corresponding strand of the DNA would have 5' end here and 3' end here. As you see, this messenger RNA has the same prime ends as coding strand of the DNA. So let me put name here. This is coding strand, coding of the DNA, and this is template strand of the DNA. So imagine that on the template strand of the DNA we have following bases. For example, cytosine, thymine, guanine, and cytosine. So what bases we are going to have on this strand of the messenger RNA? Cytosine would base pair with guanine, guanine here, and thymine would base pair with adenine, and guanine would base pair with cytosine, and cytosine would base pair with guanine, and what we are going to have on this strand of the DNA, we are going to have the same bases as on the messenger RNA. So guanine, guanine, adenine, cytosine, and guanine here. As you see, when this double-stranded DNA would return back to its initial position, when two strands of the DNA would make base pairing, this guanine would base pair with this cytosine, this guanine would base pair with this cytosine, this adenine would base pair with this thymine, and so on, with only one exception. Whenever we have, for example, adenine on the template strand of the DNA, on the messenger RNA, instead of thymine, we have uracil, and on the coding strand of the DNA, we have thymine. So, as you see, this is the only exclusion. So, this is why we call this strand of the DNA coding strand of the DNA, because the sequence, this code would be the same as in messenger RNA, with only this exception that thymine in messenger RNA would be substituted with uracil. And as you see, also 5' end here, 3' end here. In the messenger RNA and encoding strand of the DNA, we also have 5' end here and 3' end here. So now imagine that this transcription process is a part of the process in bacteria. Why? Because in bacteria, bacteria doesn't have a nucleus. So let me extend this strand of the DNA, messenger RNA, and whenever messenger RNA would be built, and here we have 5' end, this process would happen in cytosol, which has ribosomes, and instantly ribosomes would attach to this 3' end and would start a process that we call translation. So protein chain would be built. And of course, as you see, messenger RNA would grow in this direction. So it starts from here and grow in this direction continuously. So ribosome would attach to this 5' end in Shine-Dilgarna sequence. And as you know, there are 3 sites here, A site, P site, and E site. So A, P, and E. Now imagine that this is tRNA. It is not 3-dimensional, this is 2-dimensional structure. And 3-dimensional tRNA would look something like this, with Anticodon here, so we call this L form. And here we have 3' end to which amino acid would attach, and here also is 3' end, this protrusion here, and the sequence would be here A, C, C. So basically this is also RNA, this is tRNA, but still this is type of RNA. And RNA also has 2' ends. This short one would be 5' end and the long one is 3' end. So in order for tRNA to attach to specific place on this messenger RNA, which we call codon. Codon, in other words, is triplet. For example, it can be A, U, C. Then on the corresponding tRNA, we would have cytosine would base pair with guanine, uracil would base pair with adenine, and adenine would base pair with uracil. I have to put this tRNA here, but I don't have much space here to show you. So this tRNA, imagine, is taking place of the A site here. So this is going to be a sequence. Now let's pay attention to prime ends. On the messenger RNA we have 5' end here, so let's put it here, 5' end here, and 3' end here. On the corresponding tRNA we would have 5' end here, as you see, here is our 5' end, and 3' end here. If we stretch our tRNA, so imagine I stretched this tRNA, and we would have 5' end here and 3' end here. Now if I would put this tRNA here, we would have 5' end here and 3' end here. And this is exactly what happens in double-stranded DNA or double-stranded RNA. If one strand of the RNA has 3' end here, the other strand of the RNA should have 5' end here, and vice versa. So this tRNA on this picture we cannot flip or rotate as we wish. We have to put it in such a way that 5' end would be on this side and 3' end would be on the left side. So then this bases would base pair in the correct fashion. So this is going to be our tRNA here, and this is going to be long free end. This is going to be 3' end to which amino acid is attached. Now we can solve our problem. As you remember, this RNA represent this tRNA just stretched, but let's now assume that this is messenger RNA, so we have to change this prime ends. So let me change this prime ends, and if in tRNA we have 5' end here, in messenger RNA we should have a 3' end here and 5' end here. The sequence of the anticodon in our problem 3' end AUG, so we have to change the sequence on our picture to AUG. So let's change this sequence. So AUG, and on the corresponding messenger RNA we would have adenine would base pair with uracil, uracil would base pairs with adenine, and guanine would base pair with cytosine, and we would have 5' end here on the messenger RNA and 3' end here. So now the last step would be to use this table, and I on purpose brought to your attention two tables. One is DNA based and another is RNA based. So here we have codons, but codons also can be specified by coding strand of the DNA or can be specified by template strand of the DNA. So how do we know which, for example, this table whether this sequence represent coding strand of the DNA or it represent template strand of the DNA? As you remember, if this table represent coding strand of the DNA, coding sequence, then RNA sequence would be the same. So let's check. For example, we have CCG. Here we have also CCG. CCA here, CCA here, CCC here, CCC here, CCT here, and CCU here. And this is the same because in messenger RNA, thymine substitute with uracil. So as you see, the sequence in DNA and RNA table of codons is the same. So this table represents coding strand of the DNA. So these codons which we can find on the coding strand of the DNA. But for our problem we need sequence of the RNA. And here we have 5' end, 3' end, and the sequence is UAC. By the way, all these sequences here and here also are given from 5' end to 3' end. So 5' end here, 3' end here. And sequence first base is uracil, so here is our first base, second is adenine, so here is the second UA and C, so this is going to be tyrosine UAC. So this sequence on the messenger RNA corresponds with sequence AUG on the anticodon of the tRNA, and if we use RNA codon table, and this is codon table, not anticodon table, that's why we have to find first sequence of the RNA, messenger RNA. So as you see, the answer is tyrosine. So let's return to our answers, and this is answer B. This sequence in anticodon corresponds to this sequence of the messenger RNA, and according to the table, this codon specifies tyrosine. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.